These are the team members. Hopefully I'm not forgetting anybody here. Just to give some motivation for this project, we're coming at this from targeting intrinsically disordered proteins. So it's about 30% of the human proteome, and they're actually very important drug targets because about 65% of these intrinsically disordered proteins, the entire structure lacks like a distinct structure. 65% of those proteins are involved in various diseases like cancer and diabetes or like neurodegenerative diseases. And so we want to largely target this kind of undruggable part of the genome using biologics. And this isn't just restricted to human genomes, but also viral, bacterial, as well as fungal. And so the primary goals for this entire project are to, one, integrate reasoning agents to call computational biophysical tools for design, simulation, and validation. Also, we want to incorporate a RAG infrastructure to provide a high throughput literature validated hypothesis, to also scale across federated resources. That means like scale across different high performance computing resources like Aurora or Frontier. And also we want this to actually be biologically meaningful. So to actually generate biologics across different types of scaffolds like antibodies, nanobodies, affibodies, and affitins. And so this is our general approach for doing this. So let me quickly walk through this. This is a higher level overview of the co-scientist approach. And so really what this is it's using LLM reasoning as well as using a multi-agent system where you can give some research goals. So you can say, hey, I have this protein and I want to develop some kind of biologic to target it. And you can also give some preferences or constraints, saying things like, I want to focus on maybe a high binding affinity, so it needs to bind very tightly to this protein. And then this will be then sent to our multi-agent system. So this is a multi-agent system that includes various types of agents. So you would have a generation agent that can do things like explore literature and generate hypotheses. You can also have a reflection agent that can reflect on these hypotheses that you're generating, as well as evolution agents that can actually run design platforms and see if you can actually optimize the hypotheses that you're generating from the reasoner. And you can also simulate tournaments between these different hypotheses to see which hypothesis is better and which one's worse. And using this very general approach, you can actually receive some top-ranked research hypotheses and proposals that are summarized in a way that a scientist can actually incorporate into their designs. So to give a little bit of a more fine-grained view of this, and also to illustrate how we're going to be using federated resources here, let me walk through this application of this. So in this case, we're using a specific protein, NMNAT2, and you can say, okay, my prompt is I want to specify this protein target, NMNAT2, and you can go through a variety of different agents here in order to optimize the binding for this particular protein. And so in this biologic design, what we really want to do is we want to target not only NMNAT2, but also want to target certain binding sites where this protein might interact in a real biological setting. So NMNAT2, as you can see in this picture, this movie, it actually interacts with P53, which is a common cancer target. And so you would want to essentially target that interface. And so to do this in our workflow, we can take NMNAT2, this protein. This will be sent to the struct bioreasoner, which is then sent into our hyper-RAG agent. And this hyper-RAG agent, you'll query essentially, I want to find proteins that bind to NMNAT2, as well as what are the residues that bind to it. And so from this hyper-RAG agent, this will give you a hypothesis for this is the protein that binds, and these are some potential residues. This will generate some interacting sequences, and you can then send this into the biophysical, like in silico tool callings. So you can send this into this thing that we have called the Chi agent, which is basically the Chi folding model. So it's like AlphaFold, but it's a different version of AlphaFold. And this can model the interactome structure. So this is an example of the interactome structure here. You can see in this figure. And we can then take this folded structure and then send this directly into the MD agent. So MD meaning molecular dynamic simulations. And you can see in this movie that the MD agent can actually explore different types of conformations for this protein and this binding. And so this helps to, one, validate the binding site. So let's say this protein in this movie actually like flew away. That would be a bad binding site. And maybe the folding algorithm needs to be improved. So this is a way of validating that binding and also to identify which residues bind the strongest to this protein. So this is important because the interaction between NMNAT2 and P53 is the one that we actually want to target to do anything meaningful for inhibiting this protein that's for cancer. And so then following these agents, this can then be sent back into the struct bioreasoner, and we can ask it, can you generate some proposed biologics at that binding site? the consensus is struck by a reasoner, which can then generate hypotheses of proposed biologics at that binding site. And you can even ask it, like, I want to generate this type of scaffold, like an antibody or an affibody. And it'll generate those proposed biologics. And this is then sent to the Bindcraft agent, which will model those proposed biologics and also generate optimized biologics. And this uses a combination of folding algorithms and inverse folding to essentially generate better and better sequences for these biologics. This is then sent to the MD agent to evaluate the stability and the binding energy for these peptides. And so this is a small movie to show how this is working. So you can see that there's basically optimizing the peptide design for a peptide at this particular interface. And that interface is a very similar one to the one that I showed you in the previous slide. And we can then take these biologics that are optimized by this Bindcraft agent and then send this to DPO and do some more optimization. And finally, we output some proposed optimized peptides that can be sent to the user. And just to illustrate the preliminary results here of a very prototype workflow, this is showing an example that we had done a while back, where we were trying to optimize biologics for binding to epitopes. So this is specifically antibodies binding to epitopes of the RBD for the spike protein. And so here we show that using a combination of protein language models, DPO, as well as folding algorithms and molecular simulations, we can actually improve the binding affinity for these antibodies to both the wild type as well as the Omicron variant for the RBD of the spike protein. And so this is illustrating that indeed, like using all these tools in this combination, we can actually generate real biologics. But what we need now is we're developing this automated way of generating these biologics. And so future direction here is taking this platform, so this entire automated platform where we use these reasoning agents to design these biologics and then integrate this into the automated lab infrastructure that we have at Argonne. And so essentially we design those biologics and we can ask the automated experimental lab to validate these experimentally. And then we can also integrate this experimental evidence back into the design platform. So with that, I'd like to take any questions.